All right, next let's talk about modifying the basic rhythm or sinus rhythm of the heart. We can do this via extrinsic innervation of the heart, especially via innervation by autonomic nervous system neurons. Now, autonomic nervous system neurons talk to the heart through neurons that are connected to cardiac centers located in the medulla oblongata. Now, these cardiac centers include the cardioexcitatory center and the cardioinhibitory center. The cardioexcitatory center is able to send signals through sympathetic neurons to increase both rate and force of the heart. So increase both heart rate and heart contractile force. We're able to do this by stimulating the sinoatrial node and the AV node or atrioventricular node. We can also do this by stimulating the heart muscle and coronary arteries. So think of the cardioexcitatory center as kind of the gas to the heart. If we use neurons from this center to communicate with the heart, we're going to be increasing things. Whereas the cardioinhibitory center uses parasympathetic neurons or signals, usually what they use is the vagus nerve, to decrease heart rate. And this would be by inhibiting the sinoatrial node or inhibiting the atrioventricular node. And again, it's the vagus nerve, oftentimes associated with the parasympathetic nerve. So, parasympathetic system, Think of the cardioinhibitory center as the brakes. So if the cardioexcitatory center is going to send signals to the heart, we're going to speed up stuff. Whereas when the cardioinhibitory center sends signals to the heart, we're going to decrease or slow down stuff. So let's look at the autonomic innervation of the heart. Here's the medulla oblongata. So we have an implied spinal cord here. So we have descending signals. So we have a 2-neuron pathway. We're sending, for example, signals either via the vagus nerve or sympathetic neurons. The vagus nerve is working with the parasympathetic nervous system, so we would decrease impulses here or decrease functions of the heart, whereas the sympathetic neurons would speed up stuff in the heart. And I'm saying stuff just to simplify this image, but essentially sympathetic nervous system innervation would increase activities in the heart, including heart rate and contractile force, whereas parasympathetic nervous system impulses would decrease activities such as heart rate and contractile force in the heart. Next, let's look at the action potentials of contractile cardiac muscle cells. And you may be thinking, we just did this. We just looked at action potentials in the heart. We were just looking at the pacemaker cells. But remember, there are pacemaker, which were non-contractile, and contractile cardiac muscle. And we looked at the intrinsic cardiac conduction system, and we kept saying that when the signal goes through the heart, as it's going through the intrinsic cardiac conduction system, it has to spread out to those contractile cardiac muscle cells. So there's action potentials in those cells as well. Contractile muscle fibers actually make up the bulk of the heart muscle. They're responsible for the pumping action, especially of the ventricles, and they do differ from skeletal muscle contractions. The cardiac muscle action potentials have a plateau. We didn't see a plateau in skeletal muscle. If you want to go back and look at the action potential for skeletal muscle cells, go back to chapter 9. Instead, the steps involved in cardiac myocyte contractile action potentials begins with depolarization, where fast voltage-gated sodium channels open and sodium enters the cell. So not too different from skeletal muscle yet. So first step, we have depolarization, fast opening sodium channels. Once they open, we get sodium influx that makes the inside of the cell less negative. There's actually going to be positive feedback influx of sodium that causes a rising phase of the action potential as this depolarization proceeds from negative 90 millivolts to positive 30 millivolts. That depolarization by the sodium that's going into the cell is going to cause the opening of slow calcium channels. Pay attention to the slow calcium channels, because it's these slow calcium channels at positive 30 millivolts that remain open. They prolong depolarization. They create this plateau phase because at 30 millivolts, like we're used to, the sodium channels close. But in a cardiac muscle cell, unlike in a skeletal muscle cell, these slow calcium channels remain open, prolong the action potential, specifically the depolarization phase. And so it's going to create kind of longer rising phase and then postpone the falling or repolarization phase. But after about 200 milliseconds, the slow calcium channels do close. Then voltage-gated potassium channels open. We get our efflux of potassium. We get repolarization back to resting membrane potential. And the calcium that was allowed to influx for a longer period of time to create the plateau would be pumped both either to outside the cell in the extracellular space or back into the sarcoplasmic reticulum. So the difference between contractile muscle fibers and skeletal muscle fibers, if we're looking at cardiac versus skeletal, is that the action potential in skeletal muscle lasts about 1 to 2 milliseconds, super quick. Where in cardiac muscle, it lasts 200 milliseconds. That's a huge difference. The contraction in skeletal muscle lasts about 15 to 100 milliseconds as a response to that, say, 2-second action potential. Whereas in cardiac muscle, the contraction lasts over 200 milliseconds. So it also is going to be much longer of a contraction than we see in skeletal muscle. So there's benefit to this longer action potential. You may be wondering, okay, there's all these steps, but why is it important? Why does it matter? Well, first and foremost, we want to prevent tetanic contractions. We want to prevent the heart from kind of seizing up in this contracted state. And the longer refractory period granted to us by a longer action potential prevents those tetanic contractions. But also, sustained contraction ensures efficient ejection of blood. So we don't want sustained as in the heart freezes up, but we want a longer contraction so that the chambers are able to move blood efficiently. So let's review the action potential of contractile cardiac muscle cells. It begins with a depolarization. We're adding a plateau phase, which we didn't see develop in skeletal muscle action potentials. And then we'll end with repolarization. We're going to add the opening of slow calcium channels, allowing an influx of calcium from the outside of the cell. We'll also see calcium released from the sarcoplasmic reticulum, and that creates this plateau phase. That ensures a longer action potential and that the heart chambers are able to contract in a sequential manner and allow blood to move through the chambers as it's supposed to. Next, we're going to move on and look at EKGs or ECG. Same thing. An electrocardiograph actually can detect electrical currents generated by the heart, and it's actually a measure of all the action potentials in the heart at once. But it's not an action potential itself, it's an average. So it's a graphic recording of electrical activity in the heart, or a composite of all those action potentials. So it's not a single tracing, is what I'm saying. The electrodes that are used in order to take an electrocardiograph are placed at various points on the body. They measure different voltage differences, and there's usually about 12 leads, and that's probably the most typical. The main features of an EKG are a P wave, QRS complex, T wave, PR interval, ST segment, and QT interval. Read through the intervals, understand what they mean, but we're most concerned about the P wave, QRS complex, and T wave. The P wave is when depolarization of the sinoatrial node and the atria is occurring. The QRS complex is when ventricular depolarization and atrial depolarization is occurring. And the T wave is when ventricular repolarization is occurring. I just wanted to make sure to read those off so you saw that those descriptions were important, but we're going to look at everything in more detail. Here is just an image showing the electrocardiogram being given to an individual. We see the skin electrodes are being used to record an ECG. These are what are sometimes called leads. Here is a normal EKG or ECG. And what we see here are the 5 waves: P wave, Q wave, R wave, S wave, T wave, PQRST. So in order, that part's easy. P is the first bump. The QRS complex is next. And then we have the T wave is that second big bump. The reason we get the P wave, or that first bump, is because this is when the atria depolarize and contract. And so that electrical activity is picked up. The atria are smaller than the ventricles, and during the QRS complex, the ventricles depolarize and then contract. And so that's why we're seeing a much bigger wave during the QRS complex. So that's because the ventricles are depolarizing, contracting. And then during the T wave, the ventricles repolarize. So they're resetting. They're preparing to contract later. Now you may be wondering, okay, we see the atria contract, we see the ventricles contract, and then we see the bump, the T wave, for when the ventricles relax. Where's the atria relax bump? It actually gets lost in the QRS complex. The ventricles are so large, and the fact that ventricular depolarization, meaning contraction, occurs at the same time that the atria are relaxing means that we lose the bump or the wave that we would see as a separate atrial repolarization or relaxation stage. Now, if we simplify that and go through this a little quicker, first let's establish the atria contract, then relax. Then the ventricles contract and relax. Two of those things overlap. When the atria are relaxing, the ventricles are contracting. So the QRS complex is when two things are occurring. It's when the atria are relaxing and the ventricles are contracting. The P wave is what occurred first, the atria contracting. The T wave is what occurs last in those four things. It's when the ventricles relax as well. Now, I keep saying depolarization, then I say contraction, I'll say repolarization, then I said relaxation. So, are they the same thing? Essentially, you can think of them that way. So, ventricular depolarization leads to ventricular contraction. So, you can go ahead and assume depolarization means contraction. It's going to kind of make understanding the EKG a little easier. And whenever repolarization is used, go ahead and think relaxation. Because after ventricular repolarization, the cardiac myocytes begin relaxing. So let's just go through it every one more time. I'm going to sound like a broken record, but I want to make sure this is understood. The P wave occurs during atrial depolarization, or when the atria are contracting. The QRS complex encompasses both ventricular depolarization and atrial repolarization, or during the QRS complex, the atria are now relaxing, but the ventricles are now contracting. Then the T wave includes ventricular repolarization. So the atria were already relaxed, they're not yet getting ready to contract again. So the T wave is what we see when the ventricles are relaxing. The T wave is bigger than the P wave because even though the ventricles are relaxing, they're much larger than the atria. Let's go through this one more time. Atrial depolarization is shown at the P wave. And then when atrial depolarization is complete, the impulse is delayed at the AV node. So that's what we're seeing at kind of the PQ interval. Then ventricular depolarization occurs throughout the QRS complex. Atrial repolarization is also occurring. Then ventricular depolarization is complete at the ST interval. And then ventricular repolarization begins at the apex. That's what we're seeing at the T wave. And then ventricular repolarization is complete. That's what we're seeing, kind of that leveling off after the T wave. So I do recommend this image, figure 17.17, in your textbook, showing the sequence of depolarization and repolarization of the heart and how to kind of look at an EKG and its waves. Any changes in the patterns or timing of an ECG or EKG could reveal a disease or damaged heart, so a cardiovascular problem, especially something that's a problem with the heart's conduction system. Problems can easily be detected. Things like an enlarged R wave may indicate an enlarged ventricle. Elevated or depressed ST segments may indicate cardiac ischemia. So let's look at some examples. And before we move on, another problem could be a prolonged QT interval reveals a repolarization abnormality that increases the risk of ventricular arrhythmias. So we'll see some of those. Here's a normal EKG, sinus rhythm, heartbeat of about 75 beats per minute, relatively. And we can see things like multiple P waves, QRS complexes, and T waves, and they just repeat. A junctional rhythm would be when the P waves are absent, so we're missing those, but we do have a QRS complex, kind of a weird looking T wave, but this would be when the AV node kind of paces the heart. Something's wrong with the SA node, so the AV node takes over, and we get a heart rate of about 40 to 60 beats per minute. Then you have second degree heart block, so something's wrong with the AV node, and we have failure to conduct some of the SA node impulses. So as a result, there's going to be kind of more P waves. So that's what we're seeing here, kind of these extra little blips. We're going to have not as many QRS waves, and that's what we would see with second degree heart block. So something's wrong with that electrical connection between the atria and the ventricles. If something's wrong with the organization of the electrical activity, say during ventricular fibrillation, it's kind of look like something that's all over the place. Be a really chaotic EKG. This would be seen in acute heart attack or after electrical shock. So before we move on, we want to establish some terminology. We're going to end on this slide for this part of the 17B PowerPoint. When you watch the next part, make sure you understand this terminology. Systole or systole is a period of heart contraction. Diastole or diastole is a period of heart relaxation. And the cardiac cycle is blood flow through the heart during one complete heartbeat. We're going to add terminology like stroke volume and cardiac output. So it's important to at least start here and know what systole, diastole, and the cardiac cycle are. All right, so make sure you stay tuned for the next part of 17B.